students, welcome to my lesson today. Okay, I hope you're enjoying your holidays. So actually this is a revision. Actually it's a repeat of a lesson which I already had in class or in school. So by request, I've had students who wants me to do a revision, and also there are other people who want to learn about this topic as well. So with that request, so I've decided to do a lesson on this during the school holidays. Okay, so this the chapter I'm going to cover here is 6.1 and 6.2. Okay, let me see the chat box. Hello, good morning, Sawi. Good morning, Natalie. Are you able to hear me? Can you please give me feedback whether the my audio is clear? Am I able? Are you able to hear me well? Okay, so this is actually this lesson is not too difficult. The only difficult part is the development of the pollen grains and the embryo sac. Okay, I think, yeah, can hear very clearly. All right, that's good. Okay, so I will proceed with the lesson. All right, okay, so now I hope you can see this. All right, this is the 6.1, structure of a flower. So structure of a flower, there are a few parts which you have to memorize and you have to know, okay? And you need to know how to label. So the important part is whenever I give you a diagram or when the question gives you a diagram, you need to know which part it is. Okay, so very simple. All right, let's go to step by step. Now, let's look at the classification of flower first. Actually, flower, of course, flower is a plant, right? Now, let's look at the actual classification. Okay, you have actually learned chapter 8, right, for my classes. So anyway, this is more or less a backtracking lah. But when I go through this classification, because you've gone through now chapter 8, probably you will understand this. Okay, now plants is one of the kingdoms. Yeah, we have 6 kingdoms. Okay, so one of the kingdoms is called Plantae. So under plants, the characteristic is, of course, they are able to make their own food using sunlight energy. So they have chlorophyll and so on. Okay, now under plants, we have 2 major groups. Okay, so I would not want to specify whether it's phylum or whatever. Okay, I don't want to confuse you. Sometimes it's a subphylum, you know, it gets very confusing. Not just a phylum, under the phylum you have subphylum, right? Under order you have suborder. So I'm not too sure which group they are, so never mind. So under plants, actually you have 2 major groups. One is called the Bryophytes. Okay, the other one is called Tracheophytes. Now, what is the difference here? Okay, I just want to give you this extra information for you in order to understand the overall picture. I always like to give everyone an overall picture. Why are we learning flowering plants? Okay, of course, they're special, number one. Number two, also, how does it relate to entire kingdom of plants there? Okay, where does the flowering plant come here in this grouping? Okay, let's look at plants. First, we divide them into bryophytes and tracheophytes. So bryophytes are all these plants which do not have this transport system. That means don't have vascular system. Okay, they do not have the xylem, they do not have the phloem. So examples would be very small, tiny structure plants which are very small in structure, mosses and liverworts. Lumut, you call it in BM, we call it lumut. So these are without. Okay, let me write down here. So they have without vascular tissues. So they go under bryophytes. In other words, don't have xylem, don't have phloem, don't have distinct xylem, don't have distinct phloems. Okay, xylem, vascular tissues or vascular bundles. That means when I talk about vascular tissues, they refer to the xylem or phloem. Okay, the other major group here, we got a lot of the examples here under tracheophytes. So these tracheophytes, they possess vascular tissues. That means under here, you will have xylem and phloem. So xylem will be specifically for transporting water and minerals, while phloem is for transporting organic substances. That means the food and so on, sucrose, glucose, and so on. Okay, so these are vascular tissue. So with this condition, you already have 2 groups, whether with or without vascular tissues. Okay, now under tracheophytes, okay, these are with vascular tissues, you have 2 groups. Okay, now these are plants with seeds. Without seeds are called polypodiophyta. They don't have seeds. Okay, without seeds. And spermatophyta, these are plants with seeds. Okay, with seeds. So with seeds, they are actually more advanced. Okay, they're more advanced and more primitive. Primitive would be these mosses and liverworts. So you find that for those with seeds, right, with flowers and so on, they can propagate better. Okay, they are more viable in this on this earth, and there are many thousands and thousands of different species. So with more complex organization, you find that they are more able to be more, we call it sustainable. They are able to thrive better in this world, in this globe, in this on this earth. So they're actually, the numbers outweigh the numbers of the other group that do not have all these specialized tissues. Okay, so you see you've got more of vascular, those plants are tracheophytes, under tracheophytes. You also quite a lot more under the spermatophyta, and also under this with seeds, spermatophyta, we are here now. Under those with seeds, we are classified into two groups. So Gymnospermae and Angiospermae. Okay, this is family. Okay, with the AE at the back there, it's mostly of the family, or the family. Okay, the order, the we call it hierarchical family. So Gymnospermae or Gymnosperms. Okay, Gymnospermae is the classification name lah. You call about, in general name, we just call it Gymnosperms. Gymnosperms are those plants that without flowering, they do not flower. That means non-flowering. Non-flowering plants are called Gymnosperms. Okay, Gymnosperms. Examples will come under here will be those pine trees, okay, fir trees, right? Those that you mostly see overseas, but you also see in Malaysia here. Okay, those pine trees with cones, with cones that all these animals like to eat, all right? Acorns and all the cones. Okay, they are pine and fir. Now, Angiospermae or Angiosperms, they are known as. These are the ones which are flowering. Okay, flowering plants. And under flowering plants, we will have monocot and eudicot. Okay, so monocot, you have its own characteristics. Like, for example, the leaves are a bit long and then they have parallel veins. Whereas for eudicot, you have network-like veins. Okay, these are the characteristics that you know of. So here, we are looking at this whole chapter for chapter 6 is actually for flowering plants. And you'll find that because of their specialized, these are more complex, we have seed, it has seeds and it has its flowering and all that. They are more, they thrive better, okay, more sustainable. So more in numbers, and also they can live longer and so on, compared to all the other more primitive, we say more primitive plants, which are not so well developed in terms of organization. Okay, like those without seeds, those without vascular tissues. Okay, all right. So this is a general classification which I want you to understand. All right, so in order for you to get the overall picture, so we are actually looking at this lah. Now let's look at the structure. Okay, let's look at the structure of a flower. So as you know, if you have flower, all right, it definitely will produce seeds. So when a flower undergoes, or we call it pollination, they will be pollinated, okay, by the female part of the female gamete will be fertilized with the male gamete, and there will be ovule will turn into seeds, and from the seeds they grow little baby plants. Okay, the seed is just like an ovum or a zygote. It's called zygote. It's the equivalent of a zygote. Okay, which will be able to grow to get a new individual. Okay, let's look at the structure of a flower. All right, so flowers, you know that they are beautiful. All right, they serve a purpose. What purpose do they serve? So mostly flowers, they are there to attract insects, either for pollination, okay, or and some also they house the special organs. The organs are, we call gonads. Okay, so the sexual organ, flower is actually what we call the sexual organ of a plant. So whenever you see a flower, that means this plant has to be able to reproduce sexually, because this is a sexual organ for reproduction. Okay, so whenever you see got flower, means it has male, it has female. It could be in the same flower, or it could be in separate flowers. Okay, so flowers, actually there are two types. Either the flower will house both the male and female in the same structure, or you have only male flower and only female flower. Okay, so understand here that it is sexual. Whenever it's a flower, if it bears flowers, it must be able to undergo sexual reproduction. Okay, right, just like humans, right? Humans, you have the testes, you have the ovary. So these are the sexual organs. Sexual organ generally meaning the general word here is gonad. When you see the word gonad, it means sexual organ. For humans, it will be testes and ovary. For the flower, you will have the ovary and also the anther. Okay, now we look at one by one. So flowers will contain the male and the female reproductive organs, okay, which we call the gonads. Right, so where are these gonads? They are, of course, inside the flower. Okay, so in the flower, there are actually two parts. There is a male part and there's a female part. Okay, now we look at a flower here which contains both. Okay, you see, this flower here contains both the male part and the female part within the same flower. We call this flower, this type of flower is called a bisexual flower. That means got two sexes. Two sexes in the same flower. Okay, it has a male part and the female part. All right, that is called the bisexual flower. All right, bisexual flower. All right, okay, let's go on. Now, bisexual flower, that means we look at one by one. Right, for the male reproductive partner, the general name is called stamen. Just remember this, right? General name is stamen, and this is only two parts only. You need to remember, which is the anther and the filament. Okay, so simple. Just the anther. Anther, inside the anther, this is where the pollen grains are formed. Okay, anther is something like the testes. Just remember that the anther is like the testes for the human or for the mammals. Okay, anther is like the testes. And inside the testes, you have sperms, correct? Inside the testes, you have sperms. So anther, inside the anther, you will have the pollen grain. So the pollen grain is like the sperm. Okay, pollen grain is like the sperm. So here, remember, right, well, just to let you introduce to you the symbol for male. Normally, I like to write it in short form to save time. So if I want to say male, I just do this symbol. Okay, this symbol, wait, yeah, with an arrow, a circle and an arrow. All right, this is a universal symbol for male. Okay, universal symbol for male, it just shows as male. Now, how to remember? Very simple. It is like a hunter. When you see the arrow, represents a hunter, right? So males usually, traditionally, the men go out for hunting, right? They hunt and bring back the meat or bring back the animal or carcass, whatever. And the female at the home there will cook it, all right? This is caveman, right? Caveman theory, okay? Now, for the female part, the name is called carpal. Okay, carpal is the female reproductive organ, or we call it the gonad, right? The female gonad. Here, the carpal has got four parts. So you need to remember stigma. Stigma is the most obvious, that's protruding, because this stigma is supposed to catch the pollen grain. This is like the vagina, let's say, right? Vagina for the animal or for mammals. And then the style is to bring the pollen grain down, right? It is just to connect it to whatever that's down there. So this connecting to what? Connecting to the ovary, and the ovary itself contains ovule. Okay, so in terms of mammals, you have like ovary is the same, all right? You have same term in the mammals, the ovary, and inside the ovary, it produces the oocyte, or the in human or in animals, we call it oocyte or ovum, but in plants, we call it ovule. Okay, so the concept is the same. You have ovary, which produces the female gamete. So the female gamete is inside the ovule. Okay, so let me write it down here. So the female gamete. So this is the female symbol is here. All right, female, how to put a symbol? You put a circle here with an X at the bottom. So how to remember that? It's like a skirt. Remember, you imagine somebody wearing a skirt. The X there's a skirt, so it's got to be a female. Right? So this contains the female gamete. The ovule contains the female gamete, and the anther contains the male gamete. Contains, I'm not saying that is the gamete. The gamete is inside it, which I will show you later. Okay, so the male gamete is actually pollen grain. So inside here you will have pollen grain. Okay, pollen grain inside the anther. Okay, I mean here, inside here. Okay, and here the ovule is the female gamete. Okay, so the anther and the ovary are like one pair. I mean, it's like synonymous. So the male one is called anther, then the female will be the ovary. And we talk about the gamete. The gamete will be pollen grain. Okay, for this one is the pollen grain, and then for the female, it is the ovule. All right, okay. So now let's go on to how do you draw this. Oh wait, before that, let's look at the other parts. Now you have other supportive structures. These structures does not take place in any pollination or fertilization. It is only the stamen and the carpal which are the gonads. But you have other things on the flower. These other things are called the supportive structures. Supportive structures, you will have the peduncle. This peduncle actually holds the flower. It's called the tangkai bunga, or we call the flower stalk. It holds it upright, so it's able to be seen by the insect. So the wind are able to, when it blows, it can carry the pollen grain away. So it holds it in a position which is easily to be dispersed. The wind can bring the pollen grain or can be seen by the animals. Okay, then you have sepal. Now this sepal is actually something like the leaf-like structure. It is green in color. It actually covers the flower before it blooms. So before the flower blooms, I believe it opens up. Before it opens up, it's like a bud. You know, we call it a bud. So if we were to draw it, all right, I'll just try to draw it here. It's green. So this is the tangkai, which is the peduncle, right? And then you will have like this. This is the bud. Whole thing is green. This whole thing is green. All right, the flower has not come out yet. So the thing that covers it, those like leaf-like structures, they are called sepal. So once it already mature, right, going to bloom, it will grow up. So this sepal will peel backwards, and the petals will come up together with all these gonads, the structures inside. So this is actually the sepal. So once the flower is open like this, bloom already, you find the sepal much smaller. You compare in size, because the petals are much bigger. So it peels backwards. Okay, so that's the sepal. Okay, other words that you may also see will be like this. You see the word calyx and corolla. Okay, now calyx, what's the difference? Calyx is sepals. Sepal is just one piece. So if you have, let's say, 5 pieces of sepal covering the thing, all right, the whole structure is called calyx. So as you can see, it's not only one sepal. You peel here, about 5 sepals usually, okay, 5 pieces like that. You look at the base of your bunga raya. Okay, some of you will have bunga raya at home. You look at the base there of the flower, you have 5 pieces of sepal there. Okay, and corolla is referring to the structure, the whole structure of petals. Petals is only one piece, but corolla is the entire world. They call it WHORL, world. That means the whole thing. Okay, that means you will look from the top. You look at it from the top, it looks like this. From the top down, okay? From up there, look down. You will see the flower like this, like that, right? Like that, like that, like that, right? You see from the top down. So this whole thing is called the corolla. Corolla is for the petals. The calyx is for the sepal. Okay, so if you look down, also look down from the top, your sepal will look like this. 1, 2, 3, 4, 5. Okay, looking down like that. All right, so this calyx, this corolla is sitting on top of the calyx. Okay, now that is what you mean by the perianth. Okay, you don't have to be so specific. All this, whatever you don't see in your textbook, okay, forget about it. Whatever you don't see in the textbook, forget about it. Stick to your textbook. Okay, now androsium is another term just to mean that the male part, androsium. And the female part is the gynosium or gynosium, which is the carpal. Alright, so now I need to go through specifically the stamen and the carpal. So these are the 2 reproductive organs. Okay, now before that, I just mentioned they are bisexual, right? Bisexual means the flower which has 2 parts. So you can see here you have the androsium and the gynosium, which is the stamen and the carpal part, right? There are certain flowers which only have 1 organ only. I mean, either the male or female. For example, would be the papaya tree. If a papaya tree, right, if you get a papaya tree that is male, that means that plant, the papaya plant that you see, it will bloom, right? It will have flowers, but that is only the male flower. When it's a male flower, that means it only produces filament and anther. It only produces anther and a pollen grain. Okay, it does not have a carpal. But if you need to produce papaya, it means you want to eat your papaya tree, you make sure you plant a female papaya plant, you know. A female papaya plant will have the flower which has the carpal. And it's because of this carpal you can have your papaya fruit. Okay, so if you accidentally plant your papaya, happens to be a male papaya plant, you are never going to enjoy the fruits of your labor. Okay, you're not ever going to get a papaya from your male papaya tree. You have to plant a female papaya tree. Okay, but how do you know that? So sometimes you do not know because the seeds, in the seeds that you plant, let's say you plant through the seeds, right? Some of them are male, some of them are female. You wouldn't know until it starts to flower. Correct? Because it looks like a tree. Before it starts to flower, it looks normal. So you have to see when it starts to flower. Okay, if you see a female flower, okay, you're in luck. Right? You get to enjoy your papaya tree, papaya fruit. But if you happen to plant, okay, a male flower, a male papaya plant, too bad, right? You have to start over again and you have to plant a female papaya. Okay, so that's why these flowers are called unisexual. Flowers which only contain one part, they are called one either male or female, called unisexual flowers. Okay, an example of which is a papaya, right? They do not have your carpel and the stamen together in one plant. Okay, now let's start drawing. Okay, now as a bio student, you cannot run away from drawing. You need to know some question asks you to draw. So it's very simple. Do not be afraid of drawing. I'll teach you a very simple way of drawing. Okay, very simple. So first of all, you start with the carpel. Now, carpel, before I start with carpel, there are certain words sometimes you get confused. Sometimes you see the word pistil, carpel and pistil. Okay, what's the difference, carpel and pistil? A pistil is actually fused carpel. So, okay, I'm just giving additional information for you to help you, right? So, carpel is just one of this. Now, one of this is carpel. One of this, you see, looks like a vase. Okay, like a vase, or we call it a vase, that you put your flower inside. So, it looks like this long and slender thing here, and at the base of it, it is the ovary or the ovule inside the ovary. Okay, now this one. And then here you have the stigma, all right? The stigma which projects out like that, and the whole thing, that long thing's a style. Okay, so this is a carpel. This is a single carpel. If you look at this, is a single carpel, just like your bunga raya is a single carpel, because from the stamen, from the stigma down, you will see that there's style and there's only one ovary there. This is a single. Now there are certain certain different species of plants which have more than one carpel. Okay, more than one carpel. So the carpel can be fused. For example, like this one is 3 carpels. They are not fused together, right? We call it apocarpus. Okay, you don't need this. And then there's some which is like 3 carpels, but they get fused together, is syncarpus. Now, this reminds you of a fruit. Either you all think it's a vegetable. Okay, what does it remind you of? This one. When I look at this, it reminds me of my, what? Can you anyone is that? Can you guess what kind of fruit I'm trying to tell you? It's not a fruit. It's actually a fruit, not a vegetable. Okay, okay, Joanne. Yeah, I can see you, Joanne. Who is this? You cannot see her face. Okay, I put back my face. I don't know, because I'm trying to experiment a bit. Do you prefer to my face to be there? Because sometimes, I don't know, I've heard students say they prefer not to see the teacher because they say they get distracted by the teacher's face. Some people, they just want to see the bahan only. Okay, so are you saying that you want to see my face? Okay, so you just let me know, or you can personally, you can PM me. Okay, tomato is good. Yes, it looks like a tomato. Yeah, tomato. You have more than one. Okay, another one which I can think of is the capsicum. You know capsicum, the tang long jiu, or the bell pepper. Okay, you will have the middle part with the placenta, right? And then you have some seeds sticking up from the middle there. Yeah, so this could be more than one couple, which they are fused together. Okay, so in that case, if it's fused couple, we call it pistil. So here we just go to the basic one. I want to see my face. Okay, good. Right, I'll continue then. Okay, so yeah, let's continue. All right. So couple is usually talking about just that singular structure lah. Okay, so we keep it simple. We don't want to be confusing things. So let's look at the couple. Now, how to draw? All right, let's see how to draw. Very simple. You always draw like this. You want to draw the structure, you draw. Of course, it's very slippery on my glass, on the surface here. But never mind, I just try my best. You should be able to do better on your paper. So you just draw this one, all right? I'll just start with the top part here. It's always easier to draw the style first. Then you go all the bottom way, all the way to the bottom here. Then you draw something that bulges out. That to show the ovary. Okay, to show the ovary of the plant. Okay, right. A bit bengkok sorry, it's very difficult. It's too slippery. Okay, here you go and draw a little bit like this. Now, then this represents your stigma. Okay, draw a long slender neck, and then at the bottom, all right, those who are Chinese orchestra will probably think it looks like a what? Those Chinese orchestra, very long one. Don't know what's the name. It could look like a all right. Okay, right. Okay, now looks better. Okay, this is what about inside? Now you cannot be living like this. All right, inside you have to show your ovule. Now how to draw? I give you a simple way. All right, I mean besarkan this one. Very simple way of drawing it. You do it like this. Okay, then you go like this. Right, too small or too big, rather. Okay, I go like this. Right, okay. Okay, you just draw maybe. Okay, then you go this way. Okay, it looks quite nice. All right, a bit bengkok lah. I can do better on a piece of paper lah. It's too slippery, It's too slippery. Okay, like this. Maybe I draw it bigger here. Like this. How do you do like this? You go like this. Like this. Okay. Right. Okay, you can see that. Then you go like this. Okay, you can see that. Looks nice, right? And then the top part, you just join up. Right. Very nicely done. Like that. Okay, now, so you got a label. Don't forget to label. This will be the stigma. You must have 4 labels for your carpal. This is the whole thing is called carpal. So you put here your stigma. This is the style. Now, this style here is just the outer covering, Inside there are tubes, Inside there are small tubes. There's a small tube there. There's a small tube. All right, and then you have your ovary. The ovary is referring to this one. And inside the ovary, you can see the 6 there, right? Of course, there are more than that because you have different dimension on the other side. So I'm just drawing on this one flat plane. You see 6 on it. So you call it ovule. And this whole thing is known as carpal. Okay, that is your female. Now, if you want to draw the whole flower, all right? Okay, I'm sure you've done it already. Just do this. I will have to rub this off. Okay, how do I include my male part, the androecium? Okay, so different plants will have different modification. Like for the bunga raya, the anther and the filament is actually stuck at the side here. If you notice the yellow things here, this is for our bunga raya. The bunga raya, they want to come under here. They're at the side here, at the side here one. So that is for the bunga raya. There are different, of course, modifications are different. Okay, so I won't do this one here. I will follow the one you have the textbook there, which you have a very long stalk. You're going to draw a very long one. Okay, so where is the filament? Or the male part will have 2 only. So look here, the male part, you only have the anther and filament. You need to draw these 2 only. Okay, so anther and filament. How do I connect to this diagram? Okay, very simple. I just rub away this one. I rub away the names, otherwise it'll be too messy. I rub away the names first. How do I draw it? Okay, very simple. I just draw one long stalk here. Usually connect, this one connects to the base. And then here I draw the elongated kind of structure. Okay, this shows the anther. So when I want to label, you make sure you label this the anther. And then this is the filament. And collectively, these 2 is the stamen. Okay, collectively, this one is the stamen. So this is the male part. Okay, the female part we already done. Now, what about if I want to show more things? Like I want to show the peduncle, the sepal and all that. How do I draw it? Okay, simple. So you may want to make it more balanced. You can draw another one on the other side. Okay, make it more balanced. Yeah, so thick. Okay, draw another one over here. Then you can have like that. You can have like that. Okay, Okay, like that. And then how do I draw? I want to draw my sepal, I want to draw my petals and all that. Okay, no problem. So you go like this. All right, this is your peduncle. Then you will have your sepal. Okay, this will be your sepal. And then what about the petals? All right, petals, you just more or less. All right, this is one. There's another one. Okay, you'll be more complicated when you have a lot of this diagram, the names coming in. So I have to rub this off, otherwise it's difficult to read. So you can draw on another side. You can draw on just one side only. Okay, like that. Okay, then you can label this as the petal. So you're more or less done now. Okay, you have your carpal and your pistil, and then you have your other supportive structures, which would be the petal, sepal, and the peduncle. Okay, so make sure you have all these parts labeled. Always remember to label for your diagrams. If you don't label, no matter how beautiful it is, we don't give you marks. Okay, remember for biology, you have to label. And when you label, one more thing is do not put any arrows. The arrow shows progression. That means something is moving. Let's say something is coming out. Then you can use, for example, water moves out of the cell because of, let's say, osmosis. So you can draw arrow and then you can put water. Yes, you can. That is to show movement. But when you are labeling something, you want to label that inside here, let's say cytoplasm. You don't put arrow. If you put arrow, it means the cytoplasm is coming out. Okay, you just put cytoplasm like that. Remember that, that is for labeling for biology. And you do not blacken the whole thing for the nucleus. Instead, right, I think I don't know, I told you last year already. So instead, if you want to draw a nucleus, what you need to do is you have to staple. Staple means you draw dot, 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 dot. This is the correct way of drawing in a biology. Okay, and of course, you staple for the cytoplasm. So the nucleus must be more densely, the dots must be more dense, and then the cytoplasm must be less dense. See, this is a better diagram compared to this. You never label it black like that. Okay, you never do it for bio. Okay, so let's go back to our focus. So these parts all should be labeled. Now, this is how you draw. Okay, so this I show you, this will be a proper diagram. But of course, just I wrap away all this. You make sure you write all this back. Whenever you want to label, please make sure you label it properly. You label properly, ovary and ovule. And you never cross your labels. Don't ever draw, sometimes I see when you have your, let's say, diagrams like this, you never cross it. Find a way where you can put your label without crossing each other. Okay, let's say your nucleus is here. There are so many ways you can put there's so many places you can put cytoplasm. You don't need to put like this, correct? So I will circle this is wrong. You never cross your labels. Okay, and never when you write also, when you do your diagrams, right? You always, let's say you're too many things to label already. You got no more place. You don't do like this. You don't do like this and then you label it like that. You label it, let's say cytoplasm. Nobody can read like that. Nobody can read like this. For your labeling, so what you do is, you do like this, you do like this. This is better. All your labels must be horizontal. You can your diagram can be like this, L shape like that. I mean, your line can be L shape, but you cannot put your name as like, the other way. You don't put like that. You put it horizontal. Okay, so these are tips that you should know how to make your diagram more presentable to show that you are more knowledgeable about, how to draw a good biology drawing. Right? Sometimes I see this mistake, but we didn't have time to, correct it. Correct your mistakes and so on. There are many, many aspects. Okay, let's go on to next part. So we are already done. This is all you need to know. Okay, all you need to know about the parts. You need to know that your pollen grain is found in the anther. So here you can see the pollen grain when we call it 花粉. 花粉 is when the wind blows, sometimes you will get all this pollen grain being released into the air. Some people are very sensitive. They will get hay fever when it's summer or springtime. It's overseas we have spring. Here we practically every day is the same, right? The whole year is also very hot and humid. So spring fever, they call it spring fever, is when some people are very sensitive in the nose. So when during springtime, a lot of pollen grains are in the air. So when they smell, it enters they get irritated. All right, this nasal cavity is irritated, so they will have a lot of this runny nose and so on. It is actually, these things are histamine. Histamine like we learned last year. It is like irritating you is like it's like antigen. There's antigen or there's on the surface there. So it is actually irritating, is stimulating your body defense system. Your body defense system responds by producing a lot of mucus. Okay, so a lot of people with runny nose and so on. Okay, that is hay fever. Okay, because during springtime, a lot of pollen grains. So it will open. This one anther will crack. It will rupture when it's already we say mature. Okay, mature. That means every pollen grain here is ready to what they call that, to fertilize the ovule or the embryo sac, or the we call it egg cell in the ovule there. Okay, so this is the pollen grain. The purpose of your function of your anther is to produce a pollen grain. So this is the male gamete. The male gamete is inside the pollen grain, right? Okay, actually the male gamete is inside the pollen grain. The pollen grain is not the male gamete. Inside the pollen grain, there are nucleus. Now that is your male gamete. Later on we will see. Okay, for the female, you have the ovule. Inside the ovule, there is the egg cell. So that is your female gamete. Okay, right, so this is the structure, right? This is the structure of the male and female. You need to know the differences. All right, differences. Of course, the different, you see, this is lily. This is lily flower. You see the purple color one? All right, you will see that the anther is all massed already. You see it's rupturing. So it will produce this pollen grains. Okay, and the differences here you can read. Always remember, compare and contrast. When the question asks you for comparing and contrasting, always remember to give similarities and differences. At least one similarity. Otherwise, you'll be losing marks for your similarity. Okay, so you'll be losing mark because there's no similarity. Most of the people will just concentrate on your differences. But in the scheme, we normally get 1S plus 3D, for example. If the question is only 4 marks. We always allocate 1 for your similarity because you're asking to compare. Compare, that means there must be some similarity or there must be some similarity besides your differences. Okay, mention both. Okay, right. So here, how do you see this one? Just we did that for my classes. So I asked them to open up the flowers and look at the ovary and the anther and put under the microscope to see. Okay, now let's go on to the next partner. Here. I think I have an anther for you to see. Yeah, okay. Here, let's go on to 6.2 now. Development of the pollen grains and embryo sac. Now, this one gets a little bit complicated. It's not as simple as you think. But of course, like for the male, it's easier. Male is always less complicated. If you remember the spermatogenesis and your oogenesis last year from forward, okay? Spermatogenesis from one spermatogonium, which is your base, we call it the cell, the diploid cell. After it undergoes meiosis, you're going to get 4 spermatozoa, okay, which is viable. All the 4 of the sperm are able to carry out their job of fertilizing the oocyte. Okay, possible. That is for the spermatogenesis. For the male, it's the same principle. Okay, the same principle. You from one cell, which is the mother cell, we call it the basic cell. It is also going to produce 4 pollen grains. Okay, so now let's look at this. So development of pollen grains and embryo sac. So here, if you take your anther, just now I showed you the anther. Remember this one, very long one. Anther, yes, here, right? You slice it through. You slice it like this, all right, or you slice it like this. Slice it through, cross section, you will be able to see there are 4 lobes. 4, we call it 4 pollen sac. Okay, this round thing is called a pollen sac. You have 4 pollen sacs. Okay, there are 4. Okay, so whole thing is a pollen sac. Inside the pollen sac, there are called cells, which is going to be the precursor. The precursor means the origin. From this microspore mother cell, each one of them is going to further divide to get your pollen grain. So this is the puncher, or we call it the source of your pollen grain. All comes from there. Okay, so here let's look at my diagram. Okay, now my diagram I draw here. Now, so here, if you slice through, all right, now let's look at this diagram first. You slice it through, it's called a cross section. This is a cross section of your anther. you will have 4 pollen sacs. So this is one, right? The 4 pollen sacs inside there, they are filled with what we call pollen mother cells. This is the pollen mother cell, and all of them are diploid. That means, if you remember, your spermatogonium is also diploid. Okay, it's 2N. It all starts with 2N. Everything starts with the somatic cell first. So pollen sac here. Each one of these later on will undergo meiosis. Okay, so here, 4 pollen sacs. Okay, before that, let's look at this. All the layers of tissue surrounding it, they are called tapitum. The layers of the tissue surrounding it. Okay, well, we have more than one now. We have the fibrous layer, the tapitum. Tapitum is the tissue that surrounds your pollen sac. Okay, now let's look at one by one. Let's look at this one pollen sac. Just take out one only, one only. Now, I'm not looking at the whole thing. I'm just looking at this one. Just this one only. This one. Okay, I take it out. All right. Don't really take it out. Compare. I just imagine it's still there. Okay, each one of these is called a microspore mother cell. Okay, why mother cell? Because it is going to produce anak afterwards. It's going to produce some more. Okay, I take out one of this. This is your microspore mother cell. This one. Okay, next. One of these, in fact, each one of this microspore mother cell is going to, sorry, this is 2N. Right, the wrong one already. This is 2N. Mother cell is 2N. Mother cell is always 2N. It will undergo meiosis. So remember, the first process of cell division, it undergoes meiosis. And as you understand meiosis, once you finish the 2 levels of meiosis, meiosis 1, meiosis 2, you're going to get 4 cells, correct? 4 daughter cells. Okay, now these cells already become haploid. Always remember, after it completes meiosis 1, in fact, meiosis 1 already become haploid. And then after that, you have one more round, which is meiosis 2. It is still the haploid. So you have 4 haploid microspore cells. Okay, we call it a tetrad. Tetrad is a group of 4. Okay, a group of 4 is called tetrad. Now you have these things here. Okay, remember meiosis, right? Meiosis 1 and 2 lah. Of course, it always goes together. When I say meiosis, means it has to be meiosis 1, meiosis 2, because it is a whole thing, the whole set, right? Now, each one of these is now known as a microspore. Each one of these, you got 4 microspore. Okay, now 4 microspore. Not yet pollen grain, haven't formed pollen grain yet. So each one of these microspore will undergo mitosis. So the next stage is it undergoes mitosis. So you see, it also involves mitosis, not just meiosis only. It undergoes mitosis, and from one nucleus, you can see the one nucleus, it will split. You get 2 nucleus, okay, or rather 2 nuclei. Nucleus is singular. If you want to say more than that, you call it nuclei. But doesn't matter, you can say 2 nucleus, we won't mark it wrong, okay? Because we're not marking the English, we're marking your fact. So in this 2, each of this microspore, okay, let's look at 4 microspore. So each one is called a microspore. All right, it is going to actually, you get 2 nuclei. One is a tube nucleus. One is called a tube nucleus. The other one is called a generative nucleus. Okay, generative, G-E-N-E, generative nucleus. Okay, why do you need 2 types of nucleus? Okay, because the reason here, each one forms its own purpose. Each one has its own function. Now, this one, okay, I'll tell you the function later. After this, you can find this microspore will start to develop outer projections here. So we call it, the outer here will have a texture. So this is going to be the outer surface of your pollen grain. So this whole structure here, every one of them actually becomes a pollen grain. Okay, so you will have this, for usually animal or insect pollinated plants, the outer surface of the pollen grain is usually coarse, so that it sticks better onto the stigma. So usually it's coarse. For the wind pollinated, it's not so coarse. Okay, it's not as coarse as this. So, and for insect pollinated, normally the pollen grains are a bit bigger. Okay, so it's a bit heavier. For the wind one, it has to be a bit lighter so that it can be blown away carried by the wind. Okay, so I just roughly draw lah. Okay, so why you have 2? Again, your tube nucleus function here is to, when it drops onto the stigma later, it will help to generate the pollen tube. The pollen tube will bring the nucleus the male nucleus downwards to find the ovule. Okay, so like what I mean is here. It needs to go down, right? It needs to go down because the female is all the way down there. The ovule is all the way down here. Sorry, where did it go? Here. Sorry, sometimes could chat a bit. Okay, here. It has to go all the way down. So imagine the pollen grain has already landed here. Okay, the pollen grain is landed here with the male nucleus as well. How is it going to fuse with your this female gamete down here? So it needs a tube. It needs a what you call a pollen tube. So this pollen tube will grow. So we call it germinate. So it goes all the way, and then it will enter each one. Right? That each style or each one will enter one style. Okay, each pollen tube will enter one style. So you have all this will get pollinated, lah. Understand? So you need to produce a tube to carry the male nucleus down. So that is a function of your tube nucleus to germinate and produce the pollen tube. Okay, the other one is called the generative nucleus. Where's my material? Okay, here. Okay, the other one, the generative nucleus, later on during pollination, this generative nucleus will form two more nucleus, two male nucleus. One will become two later on. Okay, and these two will follow the tube down. So the tube nucleus goes down first, followed together by two male nucleus. Okay, so that will go all the way down and fertilize. Okay, and then you remember when you learn about the next one, 6.3, if you haven't seen my video, go and have a look at it later, that you have one of this will join with the egg cell, will fertilize the egg cell, you get a diploid, okay, diploid cell, and then this one will fertilize the two polar nuclei, you get a triploid, this one triploid endosperm. Okay, this one will we call it fertilize your two polar nuclei. Okay, so look at the pictures of different types of pollen grains from different species of plants. Okay, so you find that the outer structure, it is quite coarse to enable it to stick well onto the stigma. So each one of these, looking from the shape, this is all probably insect pollinated flowers, because insect pollinated one, the species, because of the structure, because it's very coarse and it's quite big. For wind pollinated, the structure of the pollen grain will be smaller, okay, and not so much texture on it. Okay, so we're done with the male part. The part is considerably easier, right? So here you just have an overall picture. You need to have an overall picture. Okay, let's look at this one. This is a picture of, you see here, this is your tetrad. So in this one, all right, this is like this. This is this one here. Then this one, each one is going to form a pollen grain. Okay, after the cell undergoes mitosis one more time, mitosis one time to get your tube nucleus and your generative nucleus. Okay, now I want to bring to your attention now. Remember my meiosis first, then only mitosis for plant. If you remember your spermatogenesis last year when you're learning about the formation of sperm, which one happens first? Mitosis or meiosis? Meiosis or mitosis? Do you remember offhand the fact for your spermatogenesis? Anyone to type in your comments here or answer here? Which one happens first for the spermatogenesis? Does mitosis happen first or meiosis happen first? Yeah, do you remember? I can start flipping your book already. Okay, Sawi, correct. Mitosis happened first. So this is a difference. Just highlighting a difference here. So how to remember? You have to find your own way. You need to remember that for the plant is the opposite for the, yes, plant is opposite from the mammals or the human. Human, you mitosis because the sporangium, right? Diploid sporangium, it has to undergo a few rounds of mitosis, and you get more and more spermatogonium, and then from there, the spermatogonium only starts your to get your spermatocyte, spermatocyte, yes, primary spermatocyte, which is also diploid. After primary spermatocyte undergoes meiosis 1, Okay, after endogous meiosis, you get spermatocyte, secondary spermatocyte, which is already haploid. Okay, remember, mitosis is first for the spermatogonium. Okay, so let's go to the female one. Female, as usual, in the oogenesis, you find is also more complicated than the male, right? So it's the same thing here with the plant kingdom, with the flowering plants. Formation of the female gamete, it's a bit more complicated. Okay, just to show you here, this is pollen tube that is germinating. Okay, we did it in school for my classes, and we were able to see actually. Now, for the thing is, remember, if you want to do this experiment, your percentage of the sucrose should be at least 10%. If you just want to see the pollen tube, you don't have to put 10% sucrose. But in order to help it to germinate, it has to be at least 10% and above. Okay, I read it somewhere. So if it's too low a concentration, the germination will be very slow. You may not be able to see at the end of your session, of your lab time. Okay, so if you're here for 3%, this is not really to see the pollen tube. If you just want to see the pollen grain, only the shape. So you can use 3% here. But if you want it to germinate, make sure you put at least 10% of sucrose solution. Okay, so this one I won't go into detail. Right, next one. Let's go to the female. The formation or the development of the female gamete. Where is the female gamete? Okay, so in female here, more complicated. You have more layers right inside here. So let's look at the terms here first. Ovules are the structures of flower formed inside the carpal. So carpal is the whole thing, including your stigma, the style, ovary. Inside the ovary, you have many, many ovules. Okay, so this picture only drawing one. So you can see it's only drawing one. In our picture here, the one I draw here has got 6. So don't think that one ovary only produces one ovule. One ovary can produce, this one I didn't show you. This is the ender, what they call the Electron microsgraph, the actual picture taken under the microscope. Okay? High magnification. Okay, now this one, the picture which I drew earlier. Okay, you can see there are at least here 6, right? On this plate, there are more which you couldn't see because it's on the other side. So remember, one ovary can have many ovules. But in order to make it simple, we just draw one. So the next picture that you see here is only one. We cannot be drawing so many, right? Too complicated. So here you see there's only one. So here, right? You can see there's only one ovule. Here, this whole thing, this whole thing is only one. You can see here, right? So here, you're only drawing one ovule. All right, now what are the names here? Nucellus, what is this nucellus? These are layers of tissue which surround the ovule. Okay, here, let's get it. Okay, first of all, the ovule attaches to the ovary wall through a stalk called a funicle. So it has to be attached to the ovary. How is it attached to the ovary? There must be some tissue to attach it. Just like a seed, when you open up a fruit, you find the seed is stuck to the ovary, correct? You have to pull it out. So that part is the funicle, and the part of the attachment a bit further up is called placenta. Just like how the baby is attached to the wall of the womb or the uterus, you have the placenta. Okay, so the placenta, as the name suggests, it supplies nutrient to the ovule, all right? It supplies nutrient. Placenta for the human as well. We supply, the human placenta supplies nutrient to the developing embryo, okay? And then a mass of tissues inside the ovary forms, developing a lump called the nucellus. Nucellus is this whole thing that has your ovule inside here, this whole thing. Okay, has your embryo sac. To be specific, it's called embryo sac. So the embryo sac, why do we call embryo sac? Because inside there got many nucleus there. Okay, later on we see how from one nucleus, how do you get 8 nucleus? Okay, it has to do with the cell division. So then after that, this nucellus, later on you get many layers of tissue. Okay, they're called integument. So one layer, okay, you can have more than one layer. We call it integument. Okay, so the next picture I show you is more clear. And then at the end of here, there's a small little hole here. It's called a micropile. This micropile is to allow, of course, the whatever to come in, the male gamete to go in, to penetrate inside, and then so it can join up or fuse together with the female gamete. Okay, it has to have a hole. And in the seed, once it already become a seed, I'm sure you've learned the parts of the seed. When you learn this seed, okay, this is the hilum, and there's a small little hole here. This is the micropile. This is the micropile where the male gamete went inside to fertilize it. Okay, this is your, what do you call that, the micropile. And also for germination, water goes into the micropile and it will break the testa. Testa is the seed coat. Once it terminates, it will break because it's going to expand. The cotyledon will expand, it breaks the testa. Testa, now what is testa? Testa is the seed coat. Where does it come from? It's actually coming from the integument. Okay, integument is before it becomes a seed, it's called integument. But after it already, what you call fertilized, it becomes a seed, right? Your integument forms the testa. The testa is the seed coat. Okay, like the biji, the selaput or the membrane on the biji there. That is called the seed coat, or it actually is called integument before that. Okay, fine, huh? So the opening is micropile, right? And then allows air and water into the seed during germination. Yeah, when it wants to germinate, it has to have water and air inside. That is the little hole that allows it to come in. Okay, now let's look at what is inside. So one of this ovule, all right, I'm going to magnify it. Here, this is another picture. I can see the nucellus. This nucellus is the whole thing. Integuments are this layer. You can see here, one layer, two layer, could be more than one. So sometimes the seed has more than one coat, seed coat. Okay, and this is the wall of the ovary. So this is the placenta. Okay, placenta. Oh no, actually this is the funicle. This one, funicle. And further up, this is the placenta. Okay, funicle, placenta. Okay, this whole thing is the nucellus. So nucellus has got many layers. You have seed coat, it covers your this whole thing. Okay, now let's look at the embryo sac. Embryo sac, mother cell. This is the one that's going to form your the egg cell. So this is called embryo mother sac, embryo sac, or megaspore mother cell. So it's either this, either you call it megaspore mother cell or embryo sac mother cell. That means these two words are interchangeable, megaspore or embryo sac. Mother cell, so it's always diploid. Always beginning starts with diploid first. Okay, so I'm drawing one here. All right, just drawing one. Okay, remember this is only one. There could be more than one inside the ovary. I'm just looking at one. Now this nucleus will undergo, this thing is going to undergo meiosis. So once it undergoes meiosis, you're going to get 4 cells as usual. Always after meiosis, you're going to get 4 daughter cells. Now out of the 4 daughter cells, as you know, same like for your oogenesis, okay, what it is going to be in unequal distribution of the cytoplasm during the cell division. The chromosomes will be equally distributed. There will be 4 sets of chromosome here. Okay, that means not 4 sets. You have N. Each one of them will be N. Okay, so the chromosome will go, you split lah. It will go to the opposite poles of the cell lah. The anaphase, remember anaphase, then metaphase first, then anaphase. So they're gonna go to different poles of the cell. Then you're gonna have N. All right, 2 times. So you're gonna have 4 daughter cells. Now these 4 daughter cells, one of it will be bigger than the rest. Okay, because most of the cytoplasm will move to one side only. So it's going to be unequal. So one is going to be slightly bigger and the others are going to be smaller, and they serve no purpose later on. It's like because it has lack of the organelles. Okay, because most of it have gone to one cell. So the others may not be able to survive. You do not have enough mitochondria, perhaps you do not have enough ribosomes, you do not have enough the organelles in order to be survivable. So you have only one. At the end of it, there's only one megaspore that is going to be useful. The other 3 will degenerate. Just same thing with your oogenesis. So these 4 are called 4 haploid megaspore. So each one is a megaspore. Out of 4, only one can carry on. One is going to be developed further. So this megaspore, all right, megaspore is already N. Once it undergo meiosis, already N. Just remember that, simple. Before meiosis is 2N. After meiosis is already N. Okay, now here, megaspore, it's going to undergo mitosis 3 times. Okay, so inside this cell, you have nuclear material. The black thing here is the nuclear material. That means the chromosomes. Okay, the chromosome is only one set. Then mitosis is just replicate only, isn't it? The number of the number of chromosomes in the daughter cells is always the same. So it just split into 2. You get first time mitosis, all right, satu kali. Then from these two groups of these daughter cells, right, actually it doesn't really split. It doesn't split cytoplasm. It's only the nuclear material split only. The is a chromosomal division only. Okay, the cytoplasm doesn't split. So chromosomal division one time. Then from these two group again split one more time. There's a second time, right? Second time mitosis, you get 4. And then another, these 4 of them split another time, 1 become 2, 1 become 2, you get altogether 8. So you have 3 times of mitosis with 8 sets of nuclear material. Okay, that means theoretically you should be able to form 8 cells. Okay, theoretically you have enough nuclear material to form 8 cells. Okay, now from here, out of the 8 of these, they are going to rearrange itself. Some of them will form a membrane around it. For example, like 3 of them will go up to the one side. We call it, it will stick up to the top there. It's got antipodal cell. So this one got membrane one. This one got membrane. Okay, it's called 3 antipodal cells. And another 2 will remain in the center without having any membrane there. They are called the polar nuclei. Okay, so I put different color for you to remember. And then another 3 will move to the opposite side of your this one. And one of it is called the egg cell, which I put in the center here. This is, the other 2 are called synergic cells. Okay, so they have different names, but you see that they are all from the one of this nucleus that started here, which is your megaspore here. Just one megaspore can form 8 nucleus inside, or 8 nuclei. From the 8 nuclei, it's going to form 3 antipodal cells, 2 synergic cells, 1 egg cell, and 2 polar nuclei. Okay, and from here, can you remember which are the ones that are actually going to take place or take part in the fertilization? Which one are going to take part in the fertilization? Okay, because this one's actually more of like revision. We've already done through that. Can you identify from this embryo sac here, which you have 8 types of cells, 8 types of, cells, structures, which one are going to actually take place in your actual fertilization? Actual fertilization. Not all 8. Some of them it's just like your, what they call the polar bodies. It will degenerate. Yeah. Egg cell, correct? Yes, egg cell, but that's not the only one. Not only this one, there is double fertilization. Don't forget double fertilization. There's another kind of fertilization. One of the male gamete is going to come here. Yes, correct. One of the male gamete will fuse with the egg cell. You will get a diploid zygote. This is going to get diploid zygote. Another male. Yes, this one. Not the antipodal. Antipodal is of no use. It's going to be like it will degenerate. It's got no use already. This one, it will fertilize your 2 polar nuclei. You will get a triploid. Triploid because the gamete is already got one end here. And then you got another end here. You got another end here. All together, you get 3 ends. It's going to be a triploid endosperm. So this one gets an endosperm. Okay, right? Okay, so this is what I want you. Yes, correct. Polar nuclei form triploid endosperm. Your egg cell will form diploid zygote. Yeah, that is your double fertilization. Okay, if you're confused, you may have forgotten your 6.3. Okay, then I might just go back to the 6.3 video and have a look at it. Okay, so here you can see this one, they only draw this one, and this one. These are the ones that take place in your double fertilization. Okay, so in 6.1 and 6.2, we just need to cover this. That's all. 6.1 is the structure, so you need to know how to draw. So I've taught you how to draw a simple one. Okay, just handle the carpal and the stamen, and label the other supporting structures. Okay, I have to tell you the difference between carpal and pistil. And over here, you need to understand the process and the progression. What becomes what. Okay, so you get from the microspore, you're going to get your pollen grain. From the megaspore, you will get your egg cell and your 2 polar nuclei from all this cell division. Okay, meiosis first, followed by mitosis. For the male, it's only one time mitosis. One meiosis, one meiosis. Okay, here. All right. not two here, the number section. So one time meiosis, one time mitosis. For here, maybe you can generate the differences and the similarities between these two. This is one of the differences. Differences is mitosis 3 times here for the female one. Okay, meiosis is 1 time, so it's similarity. So in fact, all this is very open. You can have your own, you generate your own compare and contrast points. Okay, right, any questions from here so far? I have actually done what I want to do. Okay, if any further questions, you can always contact me, okay, to ask questions. All right, okay, girls, any questions? I just wait for maybe a few more seconds so that you might be able to type something here. Okay, so it's not too difficult. It's just that you need to remember the sequence. What becomes what, it becomes what. How to remember? Read again and again and again. You will remember. If you don't read it, definitely you can remember. So read. Okay, welcome, Xiaoyi. Welcome, everyone. Okay, so I will end today's session here. Okay, I'll end today's session here. And hopefully, you will be able to understand this and get a better picture. Okay, any problem, please just ask me. All right, I'll see you in my next class. Okay, thank you, girls. Thank you, Cindy. Okay, bye-bye. I'll see you. Bye-bye.